Hi, I'm Ed Green, and I'm going to tell you a little bit about our work to identify patient-specific T-cell receptors that bind a recurrent neoepitope, based on some work in a clinical trial we've recently done. So this is based here at the German Cancer Research Center, the DKFZ. I'm going to tell you a little bit, hopefully without too much repetition, about adoptive cell therapies, the reason why we're looking for these TCRs, these T-cell receptors. Just touch very briefly on factors of the clinical trial. And this is our source of TCRs. And I understand that you've probably heard already from Neogene in one of the previous talks, so you've probably heard a lot of synthetic immunology. So I'm going to touch a little bit more on our bioinformatics processes of pulling out these TCRs from patient repertoires. But then, as this is a talk about writing the future of cancer research, we'll talk a little bit about how we're using high throughput assembly and screening of oligo libraries and how this is really helping us scale things up. So I always like to start with really the acknowledgement slide. Of course, there's a lot of people involved whenever there's a clinical trial. There's the immune monitoring unit with a fantastic set of technicians there, immune phenotyping all the patients. And then in our big boss, Michael Plattens, he's head of neurology, he runs all the lab stuff, but most of the work here is based on work from Katharina, Kwab, and Chin-Len Kyle. And I think the obligatory slide, of course, is saying if we're always looking for new and talented bioinformaticians, particularly with PhD and postdoc. So if you can code, drop me a line, I guess, in the chat afterwards. So what's a cell therapy, an adoptive cell therapy? Well, it boils down to really there's this idea of this personalized therapies, which is that you'll be a patient. We'll take out a tumour, and then through a whole bunch of either experimental or bioinformatic approaches, we'll really work out what mutations are in that tumour, and then we can use those mutations either to expand mutation-specific T cells using peptide expansion, really. So this is sort of this left arm here, where we take the patient's own cells, we expand them, then we deliver them back as a adoptive cell therapy. So Achilles Therapeutics does this in Europe, but there's plenty of players in this field. Or you can actually clone the T-cell receptor itself that seems to be binding an important neoepitope or even TAA on the tumour. Clone it, deliver it back to naive patient cells, T-cells which have not encountered the tumour, and then reinfuse those cells. So that would be here, Neogene's doing this, but this is based very much on work pioneered by Tom Schumacher in Europe and Steve Rosenberg the NIH. Now, the other idea, of course, is that you have an off-the-shelf, a non-autologous therapy. And here, you might have TCRs, you look for some that target a driver mutation, a mutation which is found in multiple patients, hopefully one that's restricted on a common HLA allele. So essentially, it's a therapeutic agent that you can deliver to multiple patients. There's a risk, to some extent, of alloreactivity, but this has been used very successfully. There's a trial, I think published now 2 weeks ago, looking at HPV, and it's a very competitive field. People are very excited by this idea. Here's sort of my slide with some of the companies involved in these sort of TCR therapeutics. So thinking now about how we ran the clinical trial, well, we're a brain tumor immunology group, and it turns out that the mutation in isocitrate dehydrogenase 1, so IDH1, it's really a driver mutation for a very particular kind of diffuse glioma. And in this particular subclass, it's present in about 70% of patients. So it's still a rare disease, but it's a recurrent target. And this mutation leads to the creation of a neoepitope, which is CD4 restricted, so on MHC2, HLA2. And so our previous preclinical work had shown that vaccinating mice against this epitope was quite successful. So the NOAA-16 trial was a multi-center, single-arm, open-label, first-in-man trial. And what was very nice about this trial was that it was very detailed immunological follow-up. So you can sort of see the trial design here, but the key point is that this third visit was when a baseline sample of blood was taken, and then throughout there were peptide vaccinations and further blood samples taken with concomitant temozolomide or chemotherapy. And here's a little schematic of The patients, I don't want to dwell on this too long because it's just been accepted in Nature, so you'll be able to read this perhaps even as this talk goes out. But the key points perhaps were that there are no detectable, at least by ELISpot, reactivity to the mutation prior to vaccination, and we could of course discuss that later, but that following the vaccination, the vast majority of patients showed a response. So that's what we're showing here. This is the visit 3, there's no response, then the responses were graded, and let's say here the majority of patients did react. So it suggests that there are T cells in there which we could fish out as a source of these TCRs binding this new epitope. And perhaps if we wanted to be generous with the data, we would say that those patients who had a stable disease or a pseudoprogression, so as it were, an immune response, likely reactivity to the tumour, they seem to show statistically significantly higher responses in the ELISpot to the vaccination. So we think, that this seems to be working. So we thought for future cell therapy, we'd want to find those TCRs. Now, how do we do this? And for classical immunology, where you know the epitope, you can do all sorts of experimental manipulations to find this. However, what I'm going to focus on a little bit here is the bioinformatics, because once you've sequenced the cells, it's essentially free. So we've created a bioinformatics stack using Docker, so it's a very scalable platform, where it doesn't really matter if you're running this on a server or your MacBook or a GPU, it doesn't matter if you're Linux or not, so all these tools are containerized. And of course we use version tracking, not only because it helps our development, but looking forward, we think this will be important going to regulators and saying, well, here's a personalized therapy, this is the exact pipeline we used on this day. So we're always trying to be forward thinking in this. We really break our tools down to sort of two groups, these sort of things which I consider to be based on the T cell genotype. So the CDR3 sequence of the TCR itself, and where you can look, and I'll touch on these later, but by either convergence selection or deep learning to see if there are any interesting patterns in these repertoires that might flag important TCRs for you. Or what we'd call sort of phenotypic analysis, so T cells, if you sequence the RNA of the TCR, of the T cells that are infiltrating a tumour, you can maybe find the activated cells directly from their gene expression profiles. There are other groups active in these fields, of course, which I've highlighted here. So just thinking now about convergence selection very briefly, TCRs with a similar CDR3 sequence, the complement determining region, are likely to bind the same epitope. So what does that look like? Well, really simplistically, if we sequence the TCRs, and this is now trivial to do with all sorts of technologies, and it's been done for a good 6-7 years now, if we characterize them, we say, well, these two seem very similar. So if we then know, well, this TCR binds this epitope, quack, it's likely that this one, because it's similar, makes the same sound, quack, but these ones here, of course, don't. Right? It's very, very simplistic, but you can see if we lay out the sequence data on top of that, we'd see, oh, look, there's a single amino acid change here, so it's very likely that this TCR still binds. In our case, what we're looking for, an IDH mutant. So we can, of course, represent that more scientifically by making a diagram of edges and nodes. So if we look at each TCR clonotype, so the number of cells expressing that TCR, we have a sort of a dot size, the node based on the frequency of that TCR, and then if we link similar TCRs with a Levenshtein distance of 1 with a node here, we can actually make very nice diagrams of this. And it's a very powerful technique because work from Alexandra Walczyk's group here, this Alice paper, has added a greater statistical understanding of the patient-specific biases and how this occurs. But focus on this diagram, you can also layer on top, is this a pre-vaccination sample, a post-vaccination sample? But I'll admit we were initially quite skeptical about this because whenever I looked at the data, there was no really good way of saying, is this really convergence selection of TCRs having a similar sequence, or is this simply sequencing error? But as the technology's developed and we've moved on to using single-cell sequencing from 10x, which is of course UMI corrected, we can prove that these things are clearly real and they are actually in the data. And so if you then look at this and you might say, oh, there's a preponderance of a GG motif, you can make a motif and use that later. A rather more recent and advanced, perhaps, method of doing this is using deep learning. And of course, the key point for deep learning is that you need a lot of data. A minimum of 500,000, I think, would be a sensible minimum. And typically you get this kind of sequencing data from the Adaptive Biosciences bulk sequencing platform, but we layer on top of that then some paired sequencing using the 10x platform so that we can recapitulate the full TCRs afterwards. So how does that look like? Well, one thing we're doing was peptide expansion assays. So you'd have your patient PBMCs, the blood cells, which you can then expand with either the mutant or the wild type peptide. And you can sequence the repertoires before in all three conditions. And then you can ask the computer just to group them. And the first thing you'll see here is that the deep learning, with really out of the box, no optimization, it can very easily group the baseline samples from the peptide expanded samples. So that was pretty surprising and quite encouraging. And we then, we looked in some more detail actually, and then we saw for some patients, whether you use the wild type or mutant peptide, the samples still group very closely together, whilst for others, here this red patient ND12, they seem to group rather distally. So we're still exploring the full significance of that. But one thing you can tell, it's a little small in this, but we had the chance to get some TILs, some infiltrating T cells, and these group dramatically differently. And the interesting thing is that each of these columns actually can be reduced to a sequence motif. So if you have a motif like this, the epitope binding part is this central region here. Using this information, you can then go back and actually screen through your large repertoires for sequences that have this motif and therefore might arguably be reacted to the new epitope. So that's what we did. But the one last method I wanted to touch on is this phenotypic recognition. So for one patient, we're actually able to acquire some material immediately post-resection. And we processed this to pull out the TILs, and we could show by ELISpot that these were tumor reactive, so at least some of them were. And then we subjected those to single-cell RNA and TCR sequencing. And you'll all be very familiar with these kind of plots, which you see in pretty much every paper. But a UMAP plot of single-cell sequencing data, and using the information from the gene expression, you can, as it were, ascertain or group these into different cell types or different types of T cells. And we spent a bit of time then finding a method to layer into this the TCR clonotype information. So the most common TCR clonotype, if this is 10,000 cells, might be present in 200-300 of those T cells. So if you map that onto here, you see, well, they tend to group in this kind of area here. And if we then sort of express that as a percentage of the cells, we'd say that, should we say 60% of the cells were in this kind of CD8 cluster. And so you'd say this is probably a certain kind of CD8 restricted, or a TCR, CD8 restricted T cell. And you can do that, of course, for all the TCRs that you have. This one here has a rather different distribution. And if you do that, we ended up making plots like this. So this was very useful for us in terms of doing our eventual candidate screening, because all of these, the top 10 TCRs in this tumor, well, the CD progression sample, they're all CD8, so there's no point actually cloning any of these, because we know that IDH is CD4 restricted. So up until this point, we were sort of doing medium to low throughput cloning. We're using double-stranded DNA fragments, and we put in a lot of work, and this is, as it were, kind of classical cloning. We developed graphical tools to facilitate the analysis of 10x and automate the process of going from single-cell data through to fragments that we want to have synthesized and cloned. We were working with cheap liquid handling platforms, had very highly optimized cloning, and very detailed SOP. So we're really churning through these things. But what I really want to sort of touch on is kind of, as we were moving into the deep learning area, we found that we need to clone really a lot of TCRs. We're getting a lot more candidates than we could manage, or at least manage affordably. So as we weren't really doing things in bulk cloning, we wondered whether we could start moving towards this oligosynthesis platform, which is available from a number of suppliers nowadays. But could we demultiplex such cheap pools where you get an enormous number of oligos but in tiny amounts? How can we do that? So the key idea here is that you have this oligo library. We have all sorts of different pieces. And these are short. Now, I think the maximum size is still about 300 base pairs. So you can flank all your sequences of interest with, as it were, primer sites, oligo sites, and then you can go into this and fish out certain oligos, in this case with the purple forward and the orange reverse primer, and this is taken from this paper here. And as you have multiple oligos, multiple components, you can then assemble them to make the full length, something that's longer than 300 base pairs. So we thought, well, which oligo should we pick? And it turns out there's a fantastic paper doing this, showing that they'd generated oligos and then did some actual cloning work, and to show that some oligos cross-reacted. So these orange spots, if you remove those, you have a pool of 166 oligos which never cross-react. So far so good, but we have to say we're a little skeptical that, I'm a kayaker, and then that Twist was telling us, we'll sell you this tube and it's more expensive than your car, or your kayaks, or your safety gear. So yeah, we thought, let's do a trial. So we looked back at the data from the paper, and I replotted the data to show the primer efficiency. If we're thinking back here, the underlying data there. And so I selected 20 primers that had a very similar efficiency. And I spiked that into an oligo library where there were 12,000 oligos doing something completely different, but there were also 96 oligos which had a forward and a reverse, and they were all different. And we didn't actually blast the library to check that these didn't bind here, but this comes up later. And then essentially we just arrayed it on a plate with forward and reverse primers, and so each combination is unique. And we said, right, does it work? And we used a tiny amount of the oligo. This was just a side experiment. You can see here we used 0.05 nanograms of the library, so this was a phenomenally small amount of DNA. And yet, even using really kind of throwaway reagents, no repeats, we showed that we can actually get all of these things. We could pick out each individual oligo of the 96. And these lines here, these are really essentially, we're working with Twist at this point, they guarantee a certain distribution of the oligo. So any dots that fall within here, this is essentially the manufacturing fidelity. And we're very happy with this actually, but we did find a couple of points where we found some doublets. So this would be a little problem with this strategy if you're trying to demultiplex with very high reliability. And so we looked into this a bit further. If you recall this, of course the nice figure in the paper underlies a rather more complex data set where in general a primer will bind only its template, but there's a certain degree of off-target. And you could say, well, I need to pick the best combination of primers so that they never cross-associate, they never bind to the wrong template. Now this is actually a very difficult problem because it's a binomial expansion, so it scales computation extremely quickly. But it did remind me of some work that NEB had done, doing a very similar sort of thing, trying to find optimal Golden Gate assembly sites, where you're saying if we're going to use TTTT, what other sites could we use to ensure that there's no off-target assembly? So we reached out to the authors of the paper, which will turn up in a moment, and they'd used a Markov chain Monte Carlo approach to solve this. Because we say if you're looking at 166 primers and you want 20 of them, that's 2E25 combinations. So essentially they used a software approach where you essentially make random sets and you iterate around them. It heuristically gets to a solution very close to the actual best. So they were very kind, they sent us the software, and we just bodged together a bit of a solution to no more than 20 minutes. And we then had a look and we started selecting primer sets. So again, primers where none of the primers should bind, as it were, bind to any of the other primers, so any of the other templates in the set. And we did this, and this is just a little throwaway figure, but we were able to show that it's actually, we feel that the efficiency, and we haven't actually been able to do this experimentally yet with the correct restrictions, but at least we can predict that the set should be better than the most recent Twist technical note here, which has now also been published. So we thought this was quite cool, and importantly, compared to this note, we can scale to very, very large numbers. So you can do over 20,000 independent sequences, I believe. So how does this now look? And this is really just bringing it to a close, and hopefully for those high throughput synthetic biology people out there, when we now sort of look at our sequences where we have moved to oligo pools, and if we have a target sequence of interest, then of course these are short sequences, so we do require, and there's a certain cost to this. Now, to assemble sequences, you'll need to have cloning sites. And there's all sorts of ways of taking these short oligos and making them into a long fragment. You can have ligase cycling reaction, all sorts of variants of Gibson or enzymatic assembly. Take your pick. Right? Interestingly, we've of course, the great thing about synthetic biology is you can barcode all your pieces, which means that, of course, you'll also need a constant primer in all your pieces. And on the very outside, we now, to demultiplex, we have a left and right primer from the sets that we've picked, which means that from our 20,000 oligos, we can individually ensure that we can, with very high fidelity, pick out one or more oligos. So, of course, if you have a target sequence that has to be made out of 3 or 4 oligos, you, of course, have the same amplification left and right primers. And one other thing we always do is that we make sure all our sequences are the same length, because it means there's no biases in the PCR. So if they're not sufficiently long, we'll always add a bit. And this, so bringing it together, if this target sequence is made of 2 components here, we can assemble those, and as I said, we put on a barcode which is relevant to the sequence and a sequencing primer site. But we've also had a lot of excitement and interesting times adding in UMIs, so that we're essentially barcoding each individual molecule. And that's become very interesting for us, not only in the TCR field, but in cloning all sorts of other stuff, but that's a story for another day. So thanks very much for your time, and I'd like to thank all the funders who've supported this work here in the clinical trial. We have a big bunch of funders, and then for some of the more synthetic biology TCR cloning side, here from the University of Heidelberg, our local state, and some local funding from personalised oncology programs and glioblastoma. So, thank you very much.